Hello, everyone, and welcome to today's Keynote webinar titled Towards Newborn Screening by Whole Genome Sequencing. This webinar is part of the 10th Annual Genetics Virtual Week, and I'm Susie Valdez of Labroots, and I will be your moderator for today's event. Today's educational web seminar is presented by Labroots. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located at the right of your screen. You can also participate by submitting as many questions you want during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Now, if you have any trouble seeing or hearing this presentation, just click on that Help Desk button located at the bottom of the screen within the navigation bar or from the lobby. I now present today's speaker, Dr. Stephen Kingsmore, President and CEO of Rady Children's Institute for Genomic Medicine, and Dr. Thomas Defay, Deputy Head at Diagnostic Strategy and Development at Alexion Pharmaceuticals. For a complete biography of our speaker, please visit the Biography tab at the left of your screen. Dr. Kingsmore, Dr. Defay, welcome to both of you. You may now begin your presentation.
Thank you very much. My name is Stephen Kingsmore. I'm going to start off and then I'm going to hand over to my colleague Tom Defay to close out the talk. I want to acknowledge from the get-go that this is a highly collaborative effort we're going to describe today, and the participating organizations are listed on the bottom of this first slide. So thanks to the many folk who are involved. Okay, let's summarize the talk and then we'll get into it. So what we will describe really for the first time is a system for delivering life-saving treatments to children at or before onset of symptoms. So today, the norm is to wait until a child has symptoms of a genetic disease to then start the process of figuring out which disease they have, and once they have a diagnosis, treatment is started. We want to completely change this paradigm so that treatment starts at or before symptom onset. In order to do that, we propose to screen newborns, up to 140 million newborns per year globally, for up to 1,000 genetic diseases, thereby accelerating diagnosis and implementing treatment. And that's shown in the blue box. There are 4 components to this. We have developed an open consortium. We're in the process now of inviting all interested parties to join us to really change how we do this worldwide. Let's set the stage by showing you newborn screening as it exists today in the United States. So almost every newborn is screened by a heel prick, as shown here, onto a drug spot, and it varies from state to state, but the cost in California is $141 newborn tested. 99.8% of newborns test negative, and that's the end of the story. But 0.2% screen positive for a condition, and in the US, about 32 diseases are screened for, because I get it to state the precise total number. This has been regarded as a triumph of public health, because for these diseases, it means there are exceptionally few cases that go untreated, and so outcomes are optimal. Why we feel the time is now to add genome sequencing is that that number has been pretty static for the last 20 years or so. So you can see that in 2005, 27 conditions were scored by newborn screening, and that rose to about 32 over the last 15 years. And that's translated in purple to going from 6,400 to 6,800 genetic diagnoses being made as a result of newborn screening. In that same time period, as you can see, the cost of decoding a genome has dropped unfathomably fast, as has the time it takes to make a genetic disease diagnosis by rapid whole genome sequencing. In addition to this, the number of diseases that we now know the molecular basis of has also increased dramatically. And as a result of this, there's a huge current gap between the number of genetic diseases that have effective therapies and those that are screened for by newborn screening. We hope to close that gap using rapid whole genome sequencing. So, why do we think that we are in a good position to do this? Well, you may not know our organization, Rady Children's Institute for Genomic Medicine, but we have been involved for the last decade or so in expanding access to rapid whole genome sequencing. And you can see some of the key numbers here in terms of what we have accomplished. The one number which is wrong on this slide is the number of states that now have a Medicaid coverage policy for rapid whole genome sequencing. That number now stands at 4. This slide is about a week old. So this is moving pretty rapidly, and as we come into an era when critically ill newborns have availability of diagnostic whole genome sequencing, the question is what's next? Because we're so aware that we're still missing many of these diseases, or we're diagnosing them too late. We have set up a 5-stage or a 6-stage program to introduce testing by genome sequencing for children with these disorders. Stage 1 is just about completed. We have validated about 350 genetic conditions, and we've developed an initial consortium of partners to be able to do this. Today we'll review the evidence of that prototype and feasibility studies. We plan to start testing this in newborns in the second half of this year. We will do that fairly slowly, only 200 newborns, to really sort out the kinks in offering this new capability. But as you can see, we plan to scale this and ultimately reach 1 million newborns sometime in 2026 and about 750 disorders screened for. So as we started to do this together, we found there were a number of challenges. The first one that really confronted us was, well, which genetic diseases should we screen for? And while the criteria for making those selections are well established, as you can see, they date back to 1968, really this had never been done over the last 20 years, 50 years, for newly known genetic diseases. And so there was this huge gap. And so over the last year and a half, we have been evaluating each disorder and each treatment to qualify them according to these criteria. And to date, we have accomplished that for about 350. Let me show you a little bit more detail about this. So the starting point for this was an information resource that we put together in partnership with Alexion over the last 2 years. And in this, we unified all of the different existing web resources that exist for providing information about genetic diseases. Hitherto, they had really never been interlinked. And in fact, some of the foundations, like a merging of disease names and nomenclature, had never been done before. We then used an expert panel and the so-called Defay method to go through every single disease and intervention and assess whether they were effective and also whether the evidence of effectiveness was strong enough to provide that information to a physician. And you can see an example on the right of the finished product for one of the disorders, and you can see the top couple of recommended treatments and interventions. This resource isn't intended to replace subspecialist opinions, but it reflects the fact that for children who are born outside of a specialist medical center with a full complement of pediatric subspecialists and super specialists, there may be critical delays in starting appropriate treatments. And this is supposed to close that gap and provide management guidance for critically ill children at time of diagnosis in an intensive care unit for one of these genetic disorders. This is very similar in concept to the type of information that's provided in traditional newborn screening shown on the left. And you can see our progress on the right in building this resource. 1,500 interventions of a total of about 10,000 were retained as indeed being effective and having evidence for effectiveness. And we are now going through this for a second time to requalify these conditions for newborn screening. That's a slightly different application area since most of the newborns will be healthy, in contrast to the original design of GTRX or Genome to Treatment, which was for diagnosis in critically ill infants. To date, we found that about 88% of the disorders we have examined have been retained for newborn screening. So that was the first challenge, and it was tractable, and we've overcome it, and we are just about finished with qualifications of disorders and are on target to have somewhere between 350 and 400 by the end of this quarter. The second big challenge was which variants should we screen for? Now, the thing about newborn screening, which is very different from diagnosis, as I showed on one of the first slides, is that 99.8% of babies test negative. And so we don't want a situation where we have false negative results. Those would be anxiety triggering for parents and unacceptable to the medical establishment. So we've been very selective in terms of which variants we should screen for. We've limited it to rare pathogenic and likely pathogenic variants in the ClinVar database. That's about 30,000 currently, but with time, this list will continue to accrue in parallel with the number of disorders that we test for over the next 5 years. But that's our starting point for which we already have preliminary data, and Tom will go through that with you in a couple of minutes. So challenge 1, picking disorders. Challenge 2, picking variants. Challenge 3, what would our data management system be to enable this to be fully scalable? And I want to point out that in general, across life science, we have a major issue, which is that our data sets are now so large that we can't dynamically refresh them. And that's really important in newborn screening. We want to be able to refresh our data for every child screened. That becomes very costly with traditional data management systems and traditional relational databases. So we chose a novel technology, a sparse array technology, for management system, one that we believe will actually screen to the 140 million individuals that we think would be screened for over the next decade. So we wanted to prove out that this system would work, and that meant solving something called the N+1 problem. It's very much what I just said, which is a practical test of how rapidly or how inexpensively you can refresh an entire data set when you've added another patient. So under the results there, we actually tested out this system. So we added a reference genome's worth of variants. It's HG002, one of the genome in a bottle samples, to 3,200 odd genomes from the 1,000 genome project that had been resequenced by the New York Genome Center. And so we're adding 4 million to 125 million, and we want to refresh the homozygote count and the heterozygote frequency for every variant in the data set. So the reason why this works so well is that the vast majority of the human genome is reference. So 99.8% of the human genome is reference. By using a sparse array format, we are only storing the non-reference positions. So when we did this with TileDB, it cost 6.6 cents to do that experiment I just described. In contrast, the typical way of doing this is to use a very large cluster, and I got to visit Genome England a few weeks ago, who have about 100,000 genomes, and for them it costs about $1 million a year to support this, and each query is $60 to $100. Contrast that, 6 cents versus $60 per query. Those will rapidly add up if we're doing millions of samples per year. So that was the third challenge solved in at least some form factor. And now I'm going to hand over to Tom, who gets the fun part, which is to show the initial results.
Thank you, Stephen. A great introduction to what we're doing and to what newborn screening by whole genome sequencing is and what it can do. I've been meeting with a number of experts in the field, and what they keep asking me is, are we ready for newborn screening whole genome sequencing technology? Do these methods have the sensitivity and specificity to work in real life? And that's what I'll address here. Another question I've been asked is, why is Alexion AstraZeneca rare disease so committed to shortening the diagnostic odyssey in rare disease? For us, people affected by rare disease and devastating conditions are inspirations and our guiding star. Our mission is to transform their lives through the development and delivery of innovative medicines, as well as through supportive technologies and healthcare services. We believe it is our responsibility to listen to, understand, and change the lives of patients and those who work tirelessly to help them. And this work is an important step towards doing just that. 50% of people who are affected by rare disease are children, and newborn screening by whole genome sequencing is a way to help those children. There are approximately 7,000 rare diseases and disorders, and most of these diseases are difficult to diagnose. Whole genome sequencing is the way to bring newborn screening to more of these diseases. And it is estimated that 30 million people in the US are living with rare disease, and we're committed to shortening the diagnostic odyssey and doing our part to help those people with treatable genetic conditions. So what is the problem we're trying to solve? Quite simply, early diagnosis. Today, diagnosing a genetic disorder is long and tedious, taking an average of 4.8 years and 7.3 specialists if the diagnosis is reached at all. Through this type of newborn screening by whole genome sequencing solution, we believe that we can transform the diagnostic odyssey from years to just days for treatable genetic disorders. So, will newborn screening by whole genome sequencing work? For starters, reminded that the first part of newborn screening by whole genome sequencing is a screen. Each infant identified as potentially having a genetic disorder will be reviewed and diagnosed by a healthcare provider. Our goal is to identify as many infants as possible that may have a treatable genetic disease. But there's another super critical aspect of newborn screening by whole genome sequencing that Stephen just referred to. Since the vast majority of babies are healthy and do not have a life-threatening genetic disease that has an effective treatment, we have to make sure that we do not have too many false positives. Remember, a positive result requires a pediatrician to call a family and let them know that their child might have a serious genetic disorder and arrange appropriate rapid follow-up. False positives mean that anxiety and adrenaline and worry and stress were needless and should be avoided. The general rule of thumb for newborn screening is that false positives should not be materially more common than true positives, and that's what we're aiming to accomplish here. This is an ambitious goal, but we feel that we can achieve it, and we'll discuss some reasons for confidence in the next slides. We've run two retrospective studies that I want to talk about here. The first of them involves patients who over the years have been diagnosed by rapid whole genome sequencing at Rady Children's Hospital. These are patients that Dr. Kingsmore's team have been working with in many cases. The rapid whole genome sequencing test at Rady is amongst the world's most advanced and costs about $8,000 per patient. Typically, it is run when the patient already has severe symptoms, though, so the average age of diagnosis is 446 days. Newborn screening by whole genome sequencing, on the other hand, will start at birth, will cost about $300 per patient, and the infant will be diagnosed in 3 to 5 days. We have years of experience contributing to the diagnosis of children in the NICU and PICU, and we're building a system that will work at a much larger scale and will require less expert intervention. Our goal at the beginning is to have a sensitivity of around 25% of the gold standard of rapid whole genome sequencing. In this case, that gold standard is represented by Rady Children's Hospital. This will be a learning system, and that percentage will go up from there as we get better at doing what we're doing. So Rady has diagnosed 2,190 affected children by rapid whole genome sequencing. For the first test that we ran, we selected a small subset of 28 diseases that had 57 diagnoses from that set of children. For this limited subset, we are very pleased to see that our sensitivity to be, on the slide, it reads 52%. I've got an update from work that was done just this weekend by Dr. Kingsmore. They're already at 55%. So this is very encouraging. In the near future, we'll be able to expand this full set of diseases and hope that we retain that level of sensitivity, and I think that we will. Most of the patients that were missed because the genetic change that was responsible for the disease was not characterized in a mainstream database and had to be found by a detailed review of the literature. We're already working to address that problem, and I'll talk about that in a couple slides. But first, let's remember that the math doesn't do justice to the human tragedy of genetic diseases in children today. Here are 4 cases that were part of that small subset of patients. Even using the rapid whole genome sequencing system at Rady Children's Hospital in San Diego and partner hospitals, we're still often chasing the horse after it's left the barn. You can see the age of diagnosis was 42 days for the first case, 21 years for the second, 166 days, and 2 years and 3 months. With this new newborn screening by whole genome sequencing system, targeted therapy could have been initiated after 3 to 5 days, either preventing disease manifestations or reducing them. So, we want to help as many children with treatable genetic diseases as we can. So how can we improve sensitivity? We're working with Genome, one of the founding partners in this consortium, for some time. They've shown us that there's a big opportunity in deep literature curation. There are millions of articles discussing the genetics of disease. Genome has developed a system to capture existing knowledge from 8 million full-text genomic articles and 2.9 million supplemental data sets. They call this solution Mastermind. So what does this mean in practice? Let's compare Mastermind to ClinVar, the gold standard resource that compiles information on known genetic variants found in people. And let me emphasize, we think ClinVar is terrific. It's a wonderful resource that we've been using as part of our system for some time. So ClinVar covers 11,325 genes, while Mastermind covers 19,864. In that set, ClinVar covers 929,000 variants, while Mastermind covers 10 times as many at over 9 million. In terms of full text articles indexed, where the information characterizing these variants is found, ClinVar references 78,000, while Mastermind references 8.2 million. That is a lot of additional information that we can then bring to bear to characterize those variants to help understand which of those variants are likely pathogenic. So last year, we at Alexion worked with Genome to supplement their offering with a curated analysis of a set of genes that we were interested in. This goes beyond the base Mastermind solution and involves curators assessing each variant and applying ACMG criteria to assess whether or not it is an actionable variant, whether or not it's pathogenic or likely pathogenic. Together, we made this information public, so all of you will be able to see these results in an advanced and helpful interface if you go look at Genomenon's mastermind. Here is a comparison, again, to that gold standard database, ClinVar. Let's look at the first gene. For ATP7B, ClinVar and Genomenon agreed on 235 variants as being actionable. There were an additional 132 variants that ClinVar also identified as actionable, but did not provide literature support for. When Genomenon went deep in the literature, they found 633 additional variants that did have literature support and met the ACMG criteria for pathogenicity. All told, there were 3.7 times as many variants called pathogenic or likely pathogenic that did have literature support. And we saw similar results with 3 other genes. Now, a lot of these variants are pretty rare. So for ATP7B and LIPA, we estimated how much each of these variants likely contributed to patients having disease. We estimated that including mastermind variants would result in 2 to 3 times as many positive patients being identified. Since we're already identifying roughly 50% of positive patients, at least in that one subset of patients, these estimates are very encouraging. And we will carry out a thorough analysis of the impact of mastermind later this year. You should also know that Genomenon is extending this analysis to cover all of the genes that we have identified as leading to conditions that have treatments available, and we're targeting to have that information available by the end of the year. So the second retrospective study is about selectivity. So we came up with a study that can be run on a large set of individuals that are unlikely to have a rare disease. That was 454,000 individuals sequenced by the UK Biobank. We ran an analysis on the same subset of 16 genes that are studied to get an insight into our selectivity. We found a very low number of potential false positives. For 9 out of the 16 genes, we observed either 0 or 1 false positive in that entire set of 450,000 people. This is a very encouraging result. And for some of you, it may be surprising. So let's look at that in a bit more detail. So a bit more about the UK Biobank. As I said before, these individuals have an age range of 40 to 69 and are in reasonably good health. Very few of these people should have a devastating disease that starts in infancy. For each of these individuals, we applied our newborn screening by whole genome sequencing process to identify those at risk for rare disease. If it's a dominant disease, to be flagged as at risk, that requires 1 positive variant per gene. And for the recessive diseases, that requires a positive variant on both copies of the gene. Since these people are unlikely to have a rare disease, this provides a great view of our likely false positive rate. If variants in a particular gene turn up too often, it may not be a good candidate for newborn screening by whole genome sequencing. Even in those cases, we have found that sometimes one specific variant is the source of the problem, and removing that one variant is able to dramatically reduce the false positives while having a very low impact on sensitivity. So, if you see a false positive in this set, what could it mean? So, first, the individual could be an undiagnosed true positive. This person may have survived with this disease into adulthood. Many of these rare diseases are difficult to diagnose, so it's possible that this could happen. Next, the individual may actually have the disease, but be getting effective treatment. These people would be treated true positives. Third, the particular variants responsible for this false positive may also have partial penetrance. And in this case, the variants may not cause disease manifestations in all people, and that's certainly something that we have seen before. Next, we will also see false pathogenicity calls. ClinVar does a great job of applying the ACMG criteria to make sure that the variants that are likely pathogenic are flagged. In some cases, there will be some errors, and those may lead to positives that we don't expect to see. And the final situation is two pathogenic variants that are on the same copy of the gene. So in our normal process for newborn screening by whole genome, we have to correct these cases, so it won't come up in normal practice. However, when we're looking at retrospective cases in the databases, we don't have access to patient samples to follow our normal practice. And so it may be that in some of these cases, two variants are on the same copy. The most common reasons for this is that they're strongly genetically linked and travel together in the population. This has not been reported too often, but for some genes, they are a large majority of the false positive cases that we see. So going into even more detail on the genes themselves. So for autosomal recessive disorder, in this first set of 16 genes, half of them were autosomal recessive. For that whole set of disorders, we found only one false positive in 454,000 individuals. Let that sink in for a moment. It's kind of incredible. This strongly suggests that we can achieve the specificity that we need for successful newborn screening by whole genome sequencing. For autosomal dominant X-linked recessive disorders, the false positive rate was not as impressive. But again, remember we're testing against 450,000 people. So that percentage of false positives is still very low. We're working through each of these examples to understand what is happening, and we have found that some of these can be corrected. This is an example of how this will be a learning system. I'd like to go into a little bit more detail on factor VIII, which is the second highest of the false positives that we see here. So first of all, pathogenic variants in this gene lead to hemophilia A. We originally found 46 subjects within the UK Biobank that were flagged as positive for factor VIII and potentially having hemophilia A. Bill Mowry on the Alexion team went and looked back at the aggregated ICD-9 and 10 codes and found that 11 patients within the UK Biobank did indeed have hemophilia A. From a sensitivity perspective, this is great. We know that the test works. It's able to pull patients that are positive for these diseases from this large data set. These should be thought of as treated true positives, and we have removed them from the false positive set. In addition, the most common variant associated with these positive individuals is known to cause only a mild version of hemophilia that would likely only have been seen in the cases of major accident or surgery. These individuals are likely partial prevalence, true positives with mild disease. We've left them in the set for now, but the choice could be made not to screen for them. All told, this analysis suggests that for this gene, the rate of false positives versus true positives will likely be acceptable. So given these encouraging results, we extended our analysis to 346 GTRX genes. Bear in mind, this set of genes was originally compiled for use in the neonatal intensive care unit, as Stephen has described. A number of these genes will be excluded from the final list and a number of additional genes added. Just as we saw with a more limited set of 16 genes, 63% of the genes showed one or no false positive in the UK Biobank set. This is really amazing specificity. At a minimum, it shows that if we limited ourselves to those genes, newborn screening by whole genome sequencing will work, and we'll be able to identify many infants at risk for rare disease while delivering very few false positives. The total false positive rate for that entire set was 0.7%, and that's not including some corrections that we've already made. At the beginning of this effort, we gave ourselves a target false positive rate of 0.3%, 3 per 1,000, so that's a little bit higher than that. However, if we only drop the 10 worst performing genes out of 346, our false positive rate drops to 0.2%, which is very good and exceeds our target. Dr. Kingsmore reminded me just a couple hours ago that if you just drop the top 5 worst performing variants from the whole set, not the whole gene, just the variant itself, you get down to a target false positive rate of 0.3% and meets our target. So we're able to retain almost all of our sensitivity while having a really sensitive test. Now, let me reiterate, this is not our final list of genes, and the analysis we did on the UK Biobank is for whole exome sequences and not the whole genome sequences. In addition, we will need to repeat this analysis on other large data sets, particularly those with more diverse populations. These results are very encouraging. More work is still needed, and we will do that work. So finally, let's return to the types of false positives that may be generated by our analysis using the UK Biobank. We did see examples of each of these types of false positives that we expected to see. The first is undiagnosed true positives. A great example that showed up in our set was the gene RYR1 responsible for malignant hyperthermia. This disease typically only presents itself when the patient has undergone anesthesia. It is likely that at least some of these patients who screen positive for this gene really do have malignant hyperthermia, but we can't know for sure. Treated true positives. This is what we discussed for hemophilia caused by mutations in factor VIII. These patients had a hemophilia diagnosis. Presumably, they are effectively treated. Partial penetrance. Some variants may not cause disease manifestations in all people or mild manifestations. As we said before, this is likely the case in factor VIII that causes milder hemophilia, and many of these people may be unaware that they have this condition. False pathogenicity call. We did identify some variants that are responsible for a large number of false positives. When we went back through the literature, the evidence in support of these variants was mixed. It seems likely that at least some of these variants are not truly pathogenic or at worst have partial penetrance. As we run newborn screening by whole genome sequence, we will flag these cases and work to correct them over time. Finally, this case that comes up just for retrospective analyses, 2 pathogenic variants on the same copy of the gene. We did find and confirm a great example of this within a gene called BTD. We found a number of positive hits against this gene involving the exact 2 same rare variants, and the odds of that happening by chance is very, very low. When we went back and examined the sequence reads directly, we were able to confirm that these 2 variants were indeed on the same copy of the gene and should be removed from the false positive list. Interestingly, this is not the only gene we've seen here, and so we're compiling some additional evidence and discussing what that means from a database analysis point of view. The UK Biobank for this work has been an amazing resource for assessing the potential of newborn screening by whole genome sequencing. I'm not sure this is the use case that they had originally intended, but they've really created a valuable resource that can be used to improve the quality of care for patients. So back to Stephen for some more discussion on how our newborn screening by whole genome sequencing will work.
Thank you, Tom. So what we've done so far is to show you how we've selected genes and how we've qualified both them and variants for newborn screening. And we've shown you some case examples of children whose lives would have been immeasurably improved had this been available at the time of their diagnosis. The last thing I want to focus on is the actual methodology. We put the entire workflow together. So this slide shows how you perform diagnostic rapid whole genome sequencing. This is a well-established workflow. We've done this over 6,000 times. It's a little bit different in this slide in as much as it's triggered by dry blood spots rather than blood samples, which is our usual specimen type. And we use the clinical features in the patient in addition to the variants that we observe in the genome to do symptom-driven interpretation, and about 1/3 of children receive a diagnosis, 2/3 are negative. Typically, we're evaluating about 4,500 genes and 10 variants are interpreted per case. Let's compare that with the workflow for newborn screening by whole genome sequencing. The first thing to note is, wow, this is very similar. There is not an awful lot of difference between what I showed you. The big differences are the context, so that we're now including newborn nurseries for the vast majority of children tested in addition to neonatal intensive care units. The second big difference is that we anticipate a true positive rate of 0.3%. It may be a little better than that, but 0.3% instead of 33%, and a false positive rate, as Tom has shown you, of no larger than 0.3%. Largely what we're doing, as Tom has shown you, is to screen initially about 30,000 disease-causing variants, all of which have entries in GTRX. They're known to be severe diseases. They're known to have effective treatments today. So the vast majority of children, as with traditional newborn screening, will receive a negative report, but for the subset, the 0.3% or so that are true positive, a diagnostic report linked to the treatment guidance provided back to the newborn electronic health record. To do this is going to take a very large team. We have really just shown you the analytic and workflow components of this effort. But in addition, we need a true village, a community of individuals and organizations to make this a reality. It takes more than just good science. It takes an awful lot of stakeholders to be satisfied that this is the right thing to do for society. Those types of organizations are listed on this slide. It's probably incomplete, but we're talking to you today because we'd like you to be involved. If you fit in one of these circles, would you reach out to us and see how you can participate in doing this? We really believe that this is going to be the future of pediatric care, but we can't do it alone. This next slide shows the initial teams. We have not locked this down just yet, but we do want to do so over the next 30 to 60 days because we're moving into initial enrollment of newborns for testing. The technical team I've already described, and between Tom and I, we have covered just about all of the organizations. Interestingly enough, Alexion is not mentioned here. I think that's a typo. The initial enrollment sites will be in Southern California, mainly in San Diego County, but also the University of Tennessee has partnered with us, and we'll be enrolling there in Memphis. Now, the nice thing about these initial enrollment sites is in San Diego, a majority of our population are Hispanic, and in Tennessee, a majority of the babies are African American. So immediately we're going to have diversity built into our enrollment. In addition, there will be 3 sites in Greece that will be starting this before the end of the year: Athens, Thessalonica, and I can never remember the third. So we'll also have international enrollment as well as in the United States. We already have some very important stakeholder groups, such as the American College of Medical Genetics and Genomics and the National Hemophilia Foundation, who are also part of our team and helping drive this and its oversight. So far, we have about 5 pharmaceutical companies, all of whom, like Alexion and AstraZeneca, make drugs for rare genetic diseases, who have joined our team and are sponsoring our enrollment. We don't want to announce them just yet, and that the initial team is still being locked down, so we anticipate there will be additional partners added to this. If you're interested, reach out to us and let us know. We've also had a lot of support from the National Institutes of Health in seeing this be done. We have funding from NIH, but hopefully that will be a problem which is shortly solved. We've also had interest from the Burroughs Wellcome Foundation, or Burroughs Wellcome Trust, and of course from We're not the only organization who are invested in this. There are several commercial testing companies, such as Semaphore, who offer panel tests for newborns. That's a little bit different from what since we are not just screening, we actually are developing a healthcare delivery of which is to deliver therapies at or before onset of symptoms. Very similar to Genome England, they are a government-sponsored entity, similar to what I've just described, and Tom has helped me with. And there are several other groups that are listed here who have very overlapping initiatives with what we are proposing. So really there is broad interest in this around the world. This is my last slide. I just want to acknowledge the very many individuals who have contributed to this. I don't have a list of the individuals at Illumina and TileDB or Genomenon, but we do have for we do have for Genomenon, we do have for Rady, Alexion, and Genomenon. Thank you very much. These are the guys who've done all of this work. And lastly, we really feel that this is the time to introduce this to the system, to change the outcome for children globally, and I invite you to join us. Thank you very much.
And thank you, Dr. Kingsmore and Dr. Defay, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's get started. Gentlemen, it looks like we already have some questions coming in from our audience. Our first question, our audience member first says, thank you for sharing your pioneering work. Can you address the socio-biomedical outcomes of diagnosis for diseases not yet having a cure or effective treatment?
So I'll take that for starters, and Dr. Kingsmore can chime in. So first of all, we're restricting ourselves to genetic diseases that do have effective treatments currently available. And we're doing this so that we can avoid that potential negative outcomes of being diagnosed for a disease for which there is no treatment yet. I will say that in time, we will also include diseases that are in the process of having clinical trials, but that will be something that will happen in a future phase, and we'll have some control from the patients about whether or not they want to participate in that. So focusing on treatments with diseases with known treatments. Stephen, do you want to add anything to that?
Yeah, I totally agree, Tom. So we might not have made that clear. Ethically, it's very worrisome to diagnose a condition if there isn't an effective and proven therapy. And the bar for that is very high. And that was the whole point of the qualification process, which is really rigorous, that we've put in place. It's not just a matter of there being a treatment available. We have to be sure that the outcome is changed as a result of starting that treatment early, at or before onset of symptoms, versus the current paradigm, which is to wait until the child is sick and gets a molecular diagnosis. And as Tom has pointed out, that sometimes takes a decade. That's clearly unacceptable, but we have to be sure that there are effective therapies that change outcomes for every condition that we test.
Thank you so much, gentlemen. Our next question, who should we contact if we're interested in helping?
Well, for starters, you should contact us. So for instance, if you send me an email, I'll triage that and send it to some additional members of our team and follow up as appropriate. As Dr. Kingsmore said, we're really looking for people who can help make this an effective solution for everyone, and we're at the beginning of that process. So your interest and participation is welcome. Send me an email to start with.
Thank you so much. And thank you to our audience for their involvement. These are great questions coming in. You mentioned the initial technical team. What about others?
So I'll say the technical team is still growing and evolving. There's another member of the technical team that I think will be probably added next week. So certainly, if you feel that you have a unique skill set or capability let us know if your company can offer something to make this happen. A lot of folks are expressing an interest, but we really want to make this an effective solution that we can deliver as rapidly as possible. So we're very interested in hearing from you. Stephen, did you want to add to that?
Nope, that sounds good, Tom. Thank you. If you didn't know, I'm actually on a cruise ship and I'm experiencing a bit of a lag. I'm offshore in Mexico. So I'm letting Tom take the first pass on the questions.
Love it. Thank you so much, gentlemen. Dr. Kingsmore and Dr. Defay, what about conditions that do not have treatment or a cure, but there are diagnosis would impact medical management recommendations, for example, treatments to avoid?
That's a really great question. And let me reiterate, we have this team of physicians who are going through the different conditions to assess whether or not it would be helped by this methodology. I think that's a really interesting example where if there's treatments that are really dangerous that we could help avoid, that's something we should consider on this list. But we're really relying on this team of experts to help do the complex assessment to make sure that we're bringing on the right ones. But I love the suggestion.
Thank you so much. And we have time for a couple.
I'll just add a little color to that.
Absolutely.
Yeah, a great example is ryanodine receptor 1s, which causes or is associated with malignant hyperthermia. And so in these patients, they're healthy until they have depolarizing anesthetics, and they develop this horrible complication and often will either die or be very seriously ill as a result of general anesthesia. That's a great example of a disorder where there is really no treatment per se, but it's something that ought to be in the medical records so that if you ever do need a general anesthetic, the anesthesiologist would be aware and would change the types of anesthetics that are used.
Wonderful. Thank you so much. A couple more questions we have time for. Are you collaborating with any other newborn screening efforts?
Well, as Dr. Kingsmore described, we are certainly in direct conversations with Genomics England and with other newborn screening efforts. The community is relatively small, and we're in tight connection with them. And our goal really here is to bring effective diagnoses to these patients. And so we really want to collaborate with whoever we can to make sure we're bringing the most effective approaches to bear.
Thank you so much. One more question. Do you expect to see such high specificity?
I have to say, I was really surprised when we got our first UK Biobank results back. I thought we were going to have to work a lot harder to get there. So we're not finished. We're not there yet. Like we said, we have additional work to do. But the level of specificity that we saw, to me, was actually really quite impressive. I think a lot of folks expected there to be much more partial penetrance that would lead to extensive false positives. And so that result, really, I thought was important and encouraging for the success of this effort.
Gentlemen, I want to thank you for your time today and for your informative presentation. Would you like to provide any closing remarks to our audience before we go?
Stephen, do you want to lead with this one?
Sure, I'd be happy to, Tom. So just to recap, what we've described today, it may not have sunk in, but it's a different way of practicing pediatric care, where instead of waiting for children to get ill with genetic disease and then starting a diagnostic odyssey, moving from subspecialist to subspecialist, what we are suggesting is that the time is now to screen for at least the subset of diseases that have highly effective therapies and where the evidence is strong that waiting for the current methods is not good enough. We want to screen for all of those conditions at birth and promptly start effective therapies. This is going to transform pediatric care, and we think that this will not just be a US phenomenon. We anticipate that most high and middle income countries in the world we'll have this system in place in some form factor by the year 2030.
And I'll simply add, this is real. The partners we've talked to are very excited. Folks are signing up. This is happening, and this is why Alexion AstraZeneca is so interested in supporting this work. Is we think the impact on patient diagnosis is going to be very high. That could really help a lot of people struggling with rare genetic disorders.
Well, I want to thank you both again for your presentation and for your exciting and important research. And before we go, I want to thank our audience for joining us today and for their interesting questions. Questions we did not have time to address or those submitted during the on-demand period will be addressed via the speaker using your contact information you provided at the registration. And this webcast can be viewed on demand for 2 years until April 21st, 2024. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Stay safe. Bye-bye.
Awesome. Thanks.
Bye-bye.